And today I'm going to be talking to you guys about classifying allergic rhinitis subjects and identifying single nucleotide polymorphisms using a support vector machine. So first, I'm just going to give a brief overview of allergic rhinitis, which is the phenotype that I studied, as well as support vector machines, which is a machine learning tool that I used. So allergic rhinitis is also known as hay fever or seasonal allergies, and the symptoms are pretty similar to that of the common cold. Though this is kind of mild, it is very prevalent among the population as it's the fifth most common chronic disease in the US. This leads to a really large productivity loss, which leads to an economic loss. So it is a pretty big problem in the US. And then as for the tool that I use to study this, I use support vector machines, which I use to do two things. One, classify individuals on the basis of having allergic rhinitis or not, and two, discovering SNPs or genes that are associated with allergic rhinitis. So how did we actually do this? Well, we did this by comparing single nucleotide polymorphisms, which is the variation in the genome. And it's the most common type of variation between two individuals. Additionally, these SNPs can be tied to a gene or not. In the example below, there's a base sequence that's ACG or AGG, where the only difference is the C or the G. This difference, this variation, is known as a SNP. Additionally, SNPs are denoted with an RS number or an RSID, which I'll talk about a little later. And then before we dive into the actual study that I did, I'm going to talk a little bit about some previous work that has been done in the field. So the most common types of studies are GWAS studies, which are genome-wide association studies, in which a researcher will take thousands of subjects and split them into two groups. They then examine their DNA that's inserted into these these microarray chips that have software that can scan for frequencies. And the computer scans a really vast area to find correlations and patterns. However, like I said, it takes thousands and thousands of subjects and can be quite costly. So I kind of proposed a way to use machine learning to do the same exact thing, which is a lot cheaper and faster. So like I said, just to reiterate, I have kind of three main points I wanted to do. One, given this genetic SNP information, I wanted to be able to classify individuals on the basis of allergic rhinitis. Second, after doing that, I wanted to see if I could examine the model and find prominent SNPs and genes associated with the onset of allergic rhinitis. Lastly, I just wanted to overall display the use of support vector machines and machine learning in general in genetic analyses. So first, we had to get some data to run this project and train my model. I took this data from the Personal Genome Project, which is a publicly available database holding genetic data, and they have multiple locations. However, I used the US Harvard site so that the data would be formatted the same, and the genetic populations may vary if it's a different country, and it may skew our control group. So as you can see on the file on the right, this is just an example of one individual's genetic file. And as you can see, it's pretty long. It has almost like 1,000,000 rows, but it only has like 4 columns. In this file as a whole, it represents an individual's genotyped DNA. This is as opposed to sequence DNA. Sequence DNA is a lot more expensive, but more comprehensive. While genotyped DNA looks at less than 1% of the DNA, but it looks at the variations. It looks at the SNPs, which is perfect because that's what we want to study. So we parsed the entire file, taking close care to look at the RSID number and the genotype. The RSID was just the unique identifier that showed what type of SNP this was, and the genotype showed the variation of that exact SNP. So now that we had the data, we had to pre-process it because the machine learning model can't just take this categorical genetic data and do something with it. We actually had to turn it into like ones and zeros. And so we did this by doing one hot encoding. This essentially just assigns a 0 or 1 based on the allele and is placed in the RSID. So we created a really large matrix, a 201 by around 55 million matrix, to store all this data of every individual. And then this took up a lot of RAM, so we had to make a sparse matrix, which essentially just removed the data holding the zeros. So this diagram just shows the process of shaping our vectors and matrices. On the very left, you can see that we have one vector representing a unique RSID of length 27. These hold all the allele combinations, AA, AT, AC, AG, TT, etc., as well as insertions and deletions. And this was where one unique RSID held information. Then we stacked this RSID with all the other possible RSIDs we found throughout all the individualist files, which ended up being around 2 million possible RSIDs that we found. So this matrix was one individual's genetic data. We then flattened this into a vector so that we could stack it on top of each other. And we had 201 participants in total, which is why the final matrix was 201 by roughly 55 million. The one-hot encoding is just represented by these ones and zeros here. So you can see that it's mostly zeros because we only denote one in a vector, in an individual's vector. So on the very left, you can see that there is a 1 in the second position. So we could pretend maybe that this individual had the AT variation of the IYRC. And then this is just filed through until the large matrix. Then to relieve RAM, we got rid of the zeros. So the matrices look something like this. Oh, sorry. Okay, sorry about that. Okay, so now we had the actual machine learning model. This is a support vector machine for my project. And what a support vector machine does is it essentially separates two classes by a hyperplane, maximizing the distance using support vectors. Now, support vectors are just the data points that are closest to the margin of the different classes. And so what a support vector machine does is it tries to create a line or a division between these two classes that can best categorize them. Our actual model used a linear kernel, which just means that we divided our data using a 2D line. We also use L2 regularization, and this means that it will minimize the non-important data points. This is as opposed to L1 regularization, where L1 would actually delete some of these data points that the machine thinks are not useful. But since our accuracy was better with L2, we can maybe hypothesize that a lot of weights or a lot of SNPs are needed in determining this boundary, and that it's a lot more complex than just being able to delete some. Additionally, our C-value was 1, and what a C-value is, it just denotes how strict the model is going to be, if it's going to classify some points wrongly but make a general line, or if it's going to be super strict. And the C-value of 1 is pretty moderate. So then, as you can see, our training set had about 100 in each group, experimental and control. And then our validation and test sets were pretty small. So once we were able to run our model, we could also analyze the weights of the model to find the significant SNPs. So we ran two-tailed z-tests to find p-values, and we had the p-value cutoff be 5 × 10 to the -8th, which is just the genome-wide significance line. This cutoff is used in GWAS studies, and even though this isn't a GWAS study, I thought it was a good line to make because we are dealing with genetic data. And so as you can see, we looked at the positive weights and the negative weights. So our final model achieved a macro F1 score and an accuracy of 0.74. This is pretty good because we're dealing with genetic data, which is very complex. So having a 0.74 accuracy is pretty good. Additionally, we found that 736 SNPs passed this genome-wide significance line, showing that they were actually significant in either the influence of allergic rhinitis or the negative influence of allergic rhinitis. The graph just shows a breakdown of the weight feature / the SNP versus the weight significance. So we were able to analyze the most prominent SNPs, and we found that there were 20 positive and 24 negative in these most prominent ones. Out of the positively correlated SNPs, 10 of them were tied to a gene, and what was fascinating was that 4 of those 10 were tied to the same gene, EVI5. And so we can probably hypothesize that EVI5 is influential possibly in allergic rhinitis. So this table just gives a breakdown of the RSID, the genotype, the p-value, and whether it was positively or negatively correlated with allergic rhinitis. The 4 arrows show the SNPs that were correlated with EVI5. So now I'm just going to give a brief rundown of some final thoughts and some possible future work. So we were able to classify individuals because we got a 0.74 accuracy, which, like I said, is pretty good. We discovered 736 genome-wide significant SNPs. And lastly, we were also able to show that SVMs and machine learning in general are useful and efficient in genetic analyses. And I say efficient because we didn't need a high number of data points. We only had 201 files. Future work could possibly be investigating certain genes that we identified in this work, as well as reproducing this with larger sample sizes to see how this could possibly affect the accuracy maybe. I'd just like to give some final acknowledgements. First to Jerry Q, my mentor, for helping me explore this project, the Summer STEM Institute for giving me this opportunity to conduct data science, as well as the IYRC reviewers for giving me really helpful feedback on my paper. Thank you.
Great. And are there any questions for Jason? maybe lucky or unlucky, you get me to ask questions, Jason. So Jason, with that database, do you know like how diverse the database is in terms of like the population? A common issue with genetics is that many databases are based purely on European white individuals who are a certain body type, certain lifestyle, certain mostly affluent, and it really kind of lacks a lot of the low income, the malnourished, the different races, ethnicities. Do you know how diverse your data set is?
Yeah, so we weren't given any of the specific information that would be like identifiable. But I do know that it was like self-submittable. So it took like the population of the US, and it was just individuals who self-selected themselves, I guess, which could be a possible downfall to the project because it's not like exactly random or a perfect representation of the US population. However, it is random in a sense that it didn't draw from any certain area. It was just anyone who wanted to submit their data.
Really like that answer. Anyone have a question? What inspired you to pursue this project?
Yeah, so um, I was really interested in genetics as a whole. Um, and I thought that if I could combine some like really powerful techniques like such as machine learning, we could find some really interesting things about it. And I had never really been introduced into machine learning until this project really. And so when I found the opportunity, I was like, I should combine machine learning and genetics and see maybe what we can do with that. And also like another really big motivator for me was reducing like the really high costs and high number of data points needed for GWAS studies. So minimizing that by combining machine learning and genetics was one of the really big motivators for me.
Wonderful. Yeah, machine learning really is the thing of the future, especially in genetics too. Basically,
Yeah.
that's what genetics is. It's just like a huge code, and you can get a machine to read a code, you can understand it. Wonderful job, Jason.